To move water 100 meters vertically, a giant tree relies on a transport pathway that is over 99.5% internal. This diagram outlines the scale of the problem. It is tempting to assume the plant pushes this water up from the soil, but as we see at the roots, root pressure typically maxes out at less than 0.1 megapascals. Furthermore, this pressure drops to zero when transpiration rates are high. It is mathematically insufficient to force fluid to the canopy. To solve this problem, we have to look in the exact opposite direction. The water is not pushed from below, it is pulled from above, driven by a large negative hydrostatic pressure, a physical tension. This tension is transmitted all the way down the water column, from the uppermost leaves directly into the roots. Sustaining this massive pull relies entirely on the cohesive properties of water molecules. They stick together tightly enough to maintain continuous liquid columns under immense mechanical stress, without snapping. The plant's vascular system does not operate as a mechanical pump. It functions as a passive vacuum engine. To understand how it generates enough force to lift a 100-meter column of liquid, we need to locate the source of the vacuum. That source is located at the extreme ends of the canopy, inside the highly intricate, branching network of veins within a single leaf. This diagram shows leaf mesophyll cells contacting empty air spaces, their walls coated in a liquid film. Water evaporates into vapor, reducing the liquid volume. The remaining fluid is drawn into microscopic pores of the cell wall. The surface bows inward, forming curved menisci. This incredibly slight physical curvature is the exact mechanical engine powering the entire upward pull of the plant. As evaporation continues, the radius of that curvature tightens. The tighter the curve, the further the fluid pressure drops into negative values. Water has a naturally high surface tension. It strongly resists this tight curvature. Its tendency to snap back to a flat surface generates a severe pulling force on the liquid directly beneath it. Multiplied across millions of microscopic pores, in thousands of leaves, this surface tension physics generates enough aggregate negative pressure to pull hundreds of gallons of water out of the soil and into the sky. Generating negative pressure is only half the problem. Transmitting that pull downward exerts a massive inward force on the plant's transport tubes. Under this much stress, standard cellular structures would instantly collapse inward. To survive this structural demand, the plant builds a specialized low-resistance plumbing system called the xylem. The conducting cells of the xylem undergo programmed death as they mature. The plant strips out all internal membranes and organelles, leaving nothing behind but empty, hollow tubes. This diagram shows both conducting tubes reinforced with thick, lignified secondary walls. Left, a trichiid. Fluid zigzags laterally through microscopic wall gaps called pits. Right, a vessel element. Shorter, wider pipes stacked end to end let water flow straight up through open grates called perforation plates. There is a strict efficiency trade-off between the two. Wide vessels provide a highly efficient, high-volume pathway with low resistance. Tracheids operate with higher resistance, forcing water to navigate through tight lateral pits. The dead, lignified architecture of both structures perfectly prevents mechanical collapse. But by creating vast, continuous columns of stretched fluid, the xylem exposes the plant to a completely different type of physics failure. Water stretched upward by 3 megapascals of tension exists in a physically metastable state. It is highly unstable. Tension alone usually isn't enough to snap the fluid column. In controlled lab settings, thoroughly degassed water requires an extreme force of over 30 megapascals to break. In nature, air seeding triggers failure. A vessel element is stretched by tension. Microscopic air bubbles are sucked through cell wall pores. Alternatively, freezing temperatures cause dissolved gases to form bubbles. Because gases cannot resist tensile forces, the bubble violently expands, severing the liquid column. This sudden expansion is an embolism, also known as cavitation. Severely dehydrated plants actively emit high-frequency acoustic shock waves. Scientists can record these sharp, distinct clicks as microscopic bubbles rapidly expand and burst internally. Once cavitation occurs, the continuity of the water column is broken, completely halting vertical fluid transport in that specific channel. Because a single air bubble can instantly knock out a transport column, relying on large, highly efficient vessels makes the plant exceptionally vulnerable to a cascading system failure. Since the xylem network is interconnected, one expanding gas bubble should theoretically expand to fill the entire tree. To prevent total failure, plants build microscopic fail-safes. Here, an expanding gas bubble fills one cell, but is halted laterally by pit membranes, pores too small for gas. The continuous water column simply detours sideways through adjacent healthy cells, entirely bypassing the blocked conduit. Conifers evolved an active mechanical valve called the torus, a thickened central pad on the pit membrane. When cavitation occurs nearby, the sudden drop in tension physically shifts the torus. It lodges securely into the circular wall thickening, perfectly plugging the hole. The massive pressure differential from the embolism itself forcefully seals the valve, securely quarantining the dangerous gas bubble inside the dead cell. The system also has physical recovery mechanisms. At night, when tension eases, trapped gases can dissolve back into the fluid. Furthermore, plants lay down new layers of secondary growth every year, guaranteeing a fresh supply of functional xylem. Dividing the plumbing into finite microscopic elements increases fluid resistance, but it is a deliberate evolutionary compromise necessary to trap embolisms and ensure continuous water delivery. The vertical transport of water requires the plant to precisely harness the physics of evaporation at the leaf, generating massive tension that pulls continuous threads of fluid upward. Sustaining this vacuum is only possible because the xylem is an intricately engineered matrix of dead, lignified cells built to withstand intense internal crushing forces. This cross-section highlights the ultimate balance of fluid dynamics. The plant carefully weighs the sheer volume of open vessels against the intense redundancy and safety of microscopic valves to survive a constant metastable state. By mastering negative pressure and cavitation control, plants have engineered a gravity-defying hydraulic network that relies on no moving parts, a system far more reliable than anything created by modern human engineering.